Hi, and welcome to the second part of Chapter 3, dealing with our organic macromolecules, the big molecules that are important for cell structure and function. We whizzed through the carbohydrates and the lipids. One thing that I did not mention last time about carbohydrates is why they are called carbohydrates. The generalized formula for a carbohydrate is CH2O, put a parenthesis around that, multiply it by N, some number. So CH2O over and over again. So for each carbon, there is a water, a hydrate, so carbohydrate. Our simple sugars that we looked at last time, like glucose, galactose, fructose, that were all structural isomers, all had the formula C6H12O6. So there are twice as many hydrogens as either carbons or oxygens. So H12O6, if we divided that by 6, we'd get H2O. There's the water. The C6, we divide that by 6, we'd get 1 carbon. So C6H12O6 is that CH2O multiplied by 6. So that's why it's a carbohydrate. We will finish Chapter 3 talking about the other two big categories of macromolecules, the proteins and nucleic acids. What is shown on this slide is a generic amino acid. There is always a central carbon called the alpha carbon. On one side will be a hydrogen atom, and on the other side is that R group, the rest of the molecule, also called a side chain. And all amino acids have that central carbon with a hydrogen, but each one will have its own unique side chain. That is the functional group, the grouping that will give it its identity and its reactivity. All amino acids also have that amino functional group, that is the nitrogen with 2 hydrogens. And on the other side of that alpha carbon, we have the carboxyl group, also called that carboxylic acid group. That is our carbon double bound to an oxygen and single bound to a hydroxyl. So there is a hydroxyl functional group, but the entire functional group is the carboxyl. We don't isolate the carbonyl and the hydroxyl apart from each other when both are attached to a carbon. This whole thing is the carboxyl group. And the amino acid is the monomer, the single unit, that we use to build the polymer that is the protein. So with complex carbohydrates, the monomer was a monosaccharide, or a simple sugar, that we used dehydration synthesis to continually add individual monosaccharides together, building those glycosidic covalent bonds to build the polymer that is the polysaccharide. With amino acids, we will once again have dehydration synthesis between the individual monomers to build the polymer. Different monomer means the name of that covalent bond we form when we add amino acids together will have a different name. And that name when we join 2 amino acids together, or when we join a single amino acid to a polypeptide chain, that bond is called a peptide bond. There are 20 common amino acids that are used in proteins, so in biological molecules. They all have the alpha carbon with a hydrogen and they have the amino end and the carboxylic acid end, and then they have their separate R groups. Now in this image, they have sort of changed the position of where we see the R groups. We're showing the carboxyl group as sticking upward as opposed to the right, like we saw in that previous slide. But the R groups are highlighted. And they are in several categories here. We have non-polar and allopathic R groups. They're all non-polar. Here, these are polar and uncharged R groups. So they're polar, they don't donate or receive hydrogens, so they're uncharged as a whole. And then over here we have positively charged R groups, we have negatively charged R groups, and we have nonpolar aromatic, and that means we have these ringed structures. It doesn't necessarily mean they have a scent to them, but they are having these circular rings. You do not need to memorize the R groups of these 20 different amino acids, but if you were to see an image of an amino acid with the R group highlighted, you should be able to determine if that R group is polar or nonpolar, if it's charged positively or charged negatively, and it would look exactly like this picture. The reason why the R groups are highlighted is because that is what gives them their identity. It also what gives them their function when they are linked together in a protein. So peptide bonds are only found between amino acids, or between one amino acid and a growing peptide chain. We won't find them in any other kind of macromolecule. So we're showing two different amino acids here. On the top, they've shown the R group is a hydrogen atom. If we look back on the previous slide, the amino acid that has a single hydrogen atom as its R group is glycine. This would be two different glycines. When we join them together, the R group would still be the hydrogen atom. That wouldn't change. The R group does not participate in the peptide bond. We want the R group to maintain its entire whole reactive identity. The peptide bond is formed between the OH of the carboxyl end of one amino acid and a single hydrogen atom from the amino end of the incoming amino acid. This is dehydration synthesis, the same type of reaction we see when we join two sugars together. The hydroxyl off one molecule, a hydrogen atom off the other. We release water as a byproduct, and then this carbon would covalently bind to this nitrogen, and what's highlighted in the blue box is the peptide bond. We have the carbonyl carbon bound to the amino nitrogen, and now this is no longer a carboxylic acid. It is a carbon double bound to an oxygen somewhere in the middle of a molecule. This is now a carbonyl carbon called a ketone because it is that keyhole in the middle of a molecule. We maintain the amino end of the first amino acid. We maintain the carboxylic acid end of the second amino acid. It's just the carboxyl end of the first, the amino end of the second that participate in dehydration synthesis to create the peptide bond. And as we build this into a true protein by adding additional amino acids, it would be the amino end of another amino acid binding to the carboxyl end of this amino acid. We only build in one direction. We don't build in both directions. This is a cartoon of a polypeptide, multiple peptide bonds between amino acids, and it happens to be bovine serum insulin. Insulin is a peptide hormone. Hormones are chemicals that are released to control various body functions. You're probably aware that insulin is a hormone that helps to control our blood sugar levels. This happens to be the insulin from bovine, which is a cow or a bull, a bovine creature. And the one protein consists of an A chain and a B chain. So one chain of amino acids all linked by peptide bonds, a second chain of amino acids all linked by peptide bonds. Between the two chains we have this two S's. Those are sulfurs. We see another pair here. Cysteine is an amino acid that contains as its R group, or in its R group, a sulfhydryl, sulfur bound to an H, an SH. And the SH sulfhydryl of this cysteine is covalently bound to the SH of the sulfhydryl on that cysteine. And this is called a disulfide bond. Covalent bonds are between the individual amino acids, and now we have a covalent bond between cysteines on completely separate chains. We have another covalent disulfide bond between the chains, and we even have a disulfide bond between two cysteine amino acids on the same amino acid chain. And this is going to be contributing to stabilizing the structure and to giving it its final three-dimensional shape that will have its very specific activity. The beta chain of hemoglobin, that iron-containing or iron-binding and oxygen-binding molecule in blood, has several chains. There are actually 4 chains. There's 2 alpha chains and 2 beta chains. Each beta chain has 147 residues, which refers to individual amino acids. 147 amino acids in length, but if we change one amino acid, we substitute instead of the glutamic acid, we substitute and get valine, we end up with sickle cell hemoglobin. Sickle cell anemia is a blood disease that causes the red blood cells, instead of being nice little donuts, they become crescent-shaped, and those crescent shapes cannot carry oxygen like they should, and they also tend to stick because they're both crescents. Instead of being nice little donuts going through our capillaries, they get stuck to each other and can cause clots and difficulties with circulation, difficulties with organ function. So instead of having a glutamic acid, also called glutamate, we change one little bit about that and it becomes valine, and that single one chain out of 147 causes a dramatic change in the hemoglobin. When we get to our chapters on how do we actually get from the genetic blueprint of DNA to these amino acids that build our proteins, we will see how that instruction manual, the DNA, if a change happens called a mutation, it can result in changing the amino acid, which would affect the functioning of that protein. This is an actual blood smear of someone with sickle cell anemia. There are some normal blood cells, but then there are some that are crescent shaped. And it's not just the cross-section that was taken. No matter what angle we looked at it, that would be a misshapen red blood cell. Secondary protein structure refers to what happens after we link those amino acids together to build a polypeptide chain. The peptide bonds, how and in what order we link amino acids, is the primary structure, the first structure. Secondary structure, we could have alpha helix and we can have beta pleated sheet. A helix is a coil, and beta pleated sheet is more like a flattened ribbon. And so depending on the R groups, that's going to determine how closely together or how far apart the individual amino acids in that chain have to remain. And we see that we get, from the amino end, we're getting hydrogen bonds to the carbonyl carbon. And that is forming the coil. Different amino acids mean we'll be spaced out differently. We're still having the hydrogen on the amino end, hydrogen bonding to the carbonyl carbon across on a different chain, but it's in a different position. The primary sequence is different, and so these secondary structures will also be different. These secondary structures are occurring within the same primary chain. We're starting here, we're going through an alpha helix region, we end up at the end, but this is all one primary sequence. This is happening within a single chain of that original peptide bonded primary structure. The tertiary structure is determined by different types of chemical reactions. The primary structure is only covalent peptide bonds. The secondary structure is only hydrogen bonds. The tertiary structure has a lot of different bonds. We can have an ionic bond between an ammonia functional group on this amino acid that's jutting into the middle and a carboxyl group that is charged on this side. We can have hydrophobic interactions where we have very hydrophobic R group on this amino acid, very hydrophobic R group on this amino acid, and hydrophobic attract each other like the hydrophobic tails on our phospholipids. We saw the disulfide bond that can happen, we saw in the insulin. So this is part of the tertiary structure. And we can have hydrogen bonds now, not just between the amino and the carbonyl carbon, but between R groups. So alpha helices and beta sheets, the hydrogen bonds are between the amino and the carbonyl carbon. In the tertiary structure, we are now getting interactions being completely separate. R groups are having hydrogen bonds. So the tertiary structure is getting to that final 3D shape of that single original primary sequence that will hold it into this position. And as long as it maintains adequate physiological conditions, will maintain this shape. Now, a situation becomes far too acidic, far too alkaline, way too hot, we can certainly disrupt some of these bonds and change that shape. But this means this is the shape it needs to be in to have proper cellular functioning. There is a fourth level of structure. We've seen the primary, which is the sequence of amino acids held together by peptide bonds. The secondary, which is the alpha helix or the beta sheets that is held together by hydrogen bonds between the amino and the carbonyl carbons. The tertiary structure, which can have disulfide covalent bonds, can have hydrogen bonds between R groups, can have hydrophobic interactions, can have ionic interactions. And that is getting that 3D shape because of the side chain, the R group interactions. The fourth level results in two separate tertiary structures coming together and then having bonds that are the same that can occur in tertiary structure. That insulin molecule we saw was two tertiary structures, the A chain of insulin and the B chain of insulin. Each had to go all the way to the tertiary level, and then those two tertiary structures were bound by those disulfide bridges into the quaternary, fourth level structure. All proteins have to get through the tertiary level. Only those proteins that rely on more than one completed chain will go to the quaternary level. Nucleic acids are called that because they were originally discovered from the nucleus of a eukaryotic cell. So nucleic, belonging to the nucleus. We now know that nucleic acids are also found in prokaryotic cells. Those are cells that do not have a nucleus, yet they still have nucleic acid. The monomer for nucleic acid is called a nucleotide. It's going to be the most complex of the monomers that we'll see. There is a nitrogenous base, meaning it is a circular structure. It has carbons in the ring, but it also has nitrogen. We have 5 different ones. DNA has 4, which would be cytosine, thymine, adenine, and guanine. RNA also has 4, but they are cytosine, uracil, adenine, and guanine. So both DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid, contain the cytosine, adenine, and guanine nitrogenous bases. Only DNA has thymine, and only RNA has uracil. And the difference between them is just here. A methyl group on thymine and just a CH on uracil. So the base is the R group that identifies which nucleotide is it. And there's only 4 different ones in each type of nucleic acid. There is a pentose sugar, so a 5-carbon sugar. In RNA, we have ribose, and in DNA, we have deoxyribose. So, this is wrong. Deoxyribose is in DNA. There's an oxygen here on this carbon and no oxygen on this carbon. But ribose should be in RNA. Let's see if I can get it to, there we go. Nope, it should be, and I'm trying to draw on a trackpad, so forgive my poor writing. See what you get for not proofreading? So, deoxyribose is called that because in ribose we have a hydroxyl group on both of the carbons, carbons 2 and 3. With DNA, we have deoxyribose because there is hydroxyl on carbon 3, but not on carbon The other component of a nucleotide is, while I have the pen out, the phosphate group. We saw the phosphate as a functional group. Phosphates are very important in nucleotides because that is going to be one of the players in our dehydration synthesis. There are hydroxyl groups on our phosphates. There are hydroxyl groups on our sugars, and that is where we will be able to obtain water as a byproduct and make a covalent linkage between the phosphate of one nucleotide and the third carbon hydroxyl on the next nucleotide. That bond is going to be called a phosphodiester bond. DNA does exist as a double helix. It's called anti-parallel because of the arrangement of the strands. On one strand, if we looked at it, it'd be phosphate to sugar to phosphate to sugar to phosphate to sugar. And it's the same on the other side, but the bases are upside down relative to each other. And that would come into play, if I can draw it with my little pen here, without getting all of the components on it. We would see it when we see all of the atoms on there, that the sugars are upside down relative to each other on each strand. So in one strand, they get linked by an oxygen. There's a carbon there. You will see better pictures of this than what I'm drawing. So the sugar here has an oxygen pointing upwards. There is an oxygen atom that will join between them. That should be to a phosphate. That's a carbon to an oxygen to the phosphate that is sticking up off the next sugar. The sugars are all with their oxygens upward. On the opposing strand, the sugars will all have their oxygens pointing downward. That is the anti-parallel structure. So the backbone is this sugar through an oxygen to phosphate, sugar to an oxygen to phosphate, and try to change the color of my pen. There we go. The base sticks up from that carbon. Just like amino acids, the R group did not participate in dehydration synthesis. The base, the nitrogenous base of our nucleotides, does not participate in the dehydration synthesis. Right? We'd have the base sticking off this one as well. I hope you're all getting a giggle out of my terrible drawings. Because our covalent bond that would release water happens between this third carbon. The carbons would be numbered 1, 2, 3, 4. The fifth carbon is sticking out of the ring. So this is the third carbon. This is the fifth carbon. And that is where the bond between two nucleotides will always happen. The bases are these rings that are shown inside, pointing inward in this double helix, like a twisted spiral staircase. So the solid portion is the phosphates and sugars, and this interior portion is where the nitrogenous bases face each other. And each strand is covalently bound to make the one strand, but between the strands is hydrogen bonds. And on each strand, there is particular bonding that has to happen. Here is a much better drawing than what I provided. This is one strand with our sugars with the oxygens pointing upward. Here is the opposing strand, the sugars with the oxygen pointing downward. And on our one nucleotide, we have our sugar, we have our phosphate, we have our nitrogenous base that's sticking up. The second one, sugar, phosphate, nitrogenous base. We have these labels of 5 with a little hash mark and 3 with a hash mark. That stands for 5' and 3'. It is a way to tell us which carbon on the sugar are we looking at. So this would be carbon 1, this would be carbon 2, this is carbon 3, this is carbon 4, and this little corner is carbon 5. The 5' end. It's prime because it's primed to interact with something. There's no further nucleotides added at that end. And it will always have the phosphate functional group. The 3' end, there's 1, there's 2, there's 3, there's 4, and there's 5. Always has the hydroxyl. This is DNA, even if it didn't tell you here. We know it's DNA because there's no hydroxyl on the second carbon, and there's thymine. Thymine is only found in DNA. It's never found in RNA. Our second strand, same order, 1, 2, 3, 4, 5. So our 5' phosphate is on the bottom end of the molecule, and our 3' hydroxyl is on the top end of the molecule. So it's running in the opposite direction. This is our anti-parallel structure. The hydrogen bonds are between the bases on opposing strands. Oh, let's get, why not magenta? Hydrogen bonds. I'm not going to spell out hydrogens. And as is the convention, the hydrogen bonds are shown as dotted lines. We have between a thymine and adenine nitrogenous base, we have 2 places where we can make hydrogen bonds. Between a guanine and a cytosine, we have 3 places we can make hydrogen bonds. Hydrogen bonds are among the weakest of the chemical bonds that we study. And so it seems at first perhaps a little counterintuitive that something as important as the spiral staircase of our DNA is held together by hydrogen bonds. That in water, if you do this, you can break them. But remember, this is facing inward in that spiral staircase, and it's not like there are 3 nucleotides. We have about 3.5 billion nucleotides in our DNA, and it's not just the hydrogen bonds that are holding it together. That is the basic molecule, and then the DNA also tightens down on itself and becomes more compact so that those hydrogen bonds are not vulnerable to attack by any sort of enzyme or chemical. Always in our DNA, thymine will be directly across from an adenine base, and guanine will always be across from a cytosine base. That's what keeps the diameter of DNA constant. If we had a thymine pairing with a cytosine, it would be difficult to get correct hydrogen bonding and would create a really narrow space in the DNA. If we had guanine binding with adenine, we'd have two double ring structures. Again, we wouldn't really be able to form correct hydrogen bonding, and we'd create a wider space. So this way, we always have the correct base pairing between the different nitrogenous bases, maintain a uniform distance and spacing in the molecule. Depending on the organism, there may be a higher proportion of adenine-thymine pairs than there are guanosine-cytosine pairs, or there could be a higher proportion of guanine-cytosine pairs to adenine-thymine. But if you know the percentage of any single nucleotide, you can determine the percentage of all the others. So if there's 20% thymine in a particular cell's DNA, then there has to also be 20% adenine, because there has to be the exact same amount. They will always base pair with each other. So 20% thymine plus 20% adenine, that's 40%. That leaves 60% of the DNA composed of guanine and cytosine. And half of that is each, so if 60% is G plus C, then G and C are each 30%. So if you know the percentage of any one, you can calculate the percentage of the other three. Ribosomes are made of ribosomal RNA, which is a nucleic acid, and of proteins. And there is a large subunit and a small subunit. In prokaryotes, they are smaller and made of different RNAs and proteins than in eukaryotes, but they carry out the same function. So this image says, how does protein synthesis work no matter the kind of cell? We have the ribosomal RNA that composes our ribosomes. We have tRNA, which stands for transfer. It is a type of RNA that will deliver amino acids to the ribosome. And we have mRNA that stands for messenger. It actually contains the instructions in the form of ribonucleotides that tells all of this how to put this protein together. So the messenger RNA tells the tRNA, hey, you need to deliver this amino acid. The tRNA brings it to the ribosome links the amino acids that are attached to the tRNA into a growing peptide chain. And that's about a 30-second lecture of what will take up a full lecture later in the semester on protein synthesis. But our DNA is read by enzymes to create messenger RNA, also creates the T and the rRNA, but the messenger RNA contains the codes, the codons, the set of 3 nucleotides that tells the tRNA which amino acid to bring in. The tRNA's brought the amino acid, and the ribosome helps to connect those amino acids into a peptide chain. That is our whirlwind tour of the macromolecules. If you go into our module, you will be able to see a practice for doing our macromolecules. In our module, I tried to have this up, but you get to scroll. I've got a functional group practice. Parts of it weren't bringing up the actual drag and drop molecules, but it still was a nice quiz on recognizing the functional groups and their properties. And I also have this worksheet for organizing and thinking about the different types of molecules we've talked about. Now, it's not due for a grade, but I will post the key to that in a couple of days so that you can check yourself on how well you did on the macromolecule review. This week's lab is a toughie. It takes a little more time and working with the graphical analysis tool. That graphing tool is not difficult, but it does take a little getting used to. That's why I posted those screenshots for you. So remember to post your initial comment by 11:59 a.m. on this lecture, and then return before 11:59 p.m. on Wednesday, September 2nd, to reply to at least 2 of your classmates. People did a really impressive job the first week. We're going to have this exact same setup every single week for the rest of the semester, so you know exactly what to expect. And we are going to keep rolling and keep supporting each other. If you need extra help, I'm going to be posting a link to a peer tutor program that's in effect this semester. We've got some students who have done quite well in the courses in biology and chemistry, so they are going to be available for individualized help. And you're always welcome to let me know if you want to just have an email chat or set up a Zoom chat to help you out with anything you're having difficulties with. So take care, have a great week, and you'll be seeing me soon.